How are you? I hope you all are doing good. This is Sai Anusha and welcome to my channel Anu Biology Book. Thank you so much. I'm very happy that we have received a immense positive response for my first video on structure of the heart, and we have received all your comments, compliments and your valuable suggestions, and I promise you that for each and every next video we will become more and more better. Actually, we have received many suggestions and requests from students of 10th standard to explain about the topic mitosis. We all know that this is a very important topic in 10th class, and many students feel so much confused and they feel so much difficulty in understanding this mitosis. So I have decided to help all the students who are presenting their board examinations. And today, let us get into the topic of mitosis. Actually, we have done another video on the basics of cell division. If you can understand the basics of cell division, you can understand this mitosis very well. And we'll post that links very soon. If you want to watch that, you can also go through that. And now, without any delay, let's get into the topic called mitosis. So here, before going into mitosis, let me give a brief introduction about a cell. We all know that cell is the basic structural and functional unit of all living organisms, right? So if you take a cell, there are two types, prokaryotic cell and eukaryotic cell. Karyo means nucleus, pro means primitive. So this kind of prokaryotic cells do not have a particular type of nucleus or nuclear membrane. So the genetic material is not at all enclosed by a specific nuclear membrane. So if you take a eukaryotic cell, U means the advanced, the true, right? So here, coming to the eukaryotic cells, these eukaryotic cells, U means true, karyo means nucleus. So these eukaryotic cells contain the genetic material enclosed in a specific nuclear membrane, and all the cell organelles are completely covered with membrane, right? So now today we are going to see the cell division that takes place in a eukaryotic cell, that also an animal cell. Why I'm taking an animal cell means animal cell and plant cell undergo mitosis, but only a slight difference is there, not much. So if you can understand this animal cell, the plant cell is also obviously will be the same except a few differences. So now let us see the mitosis. Okay, so here before going into mitosis, I want to say a few points about a cell. So if you take a cell, a human cell contains basically a few type of components, very important components. The first important component is cell membrane. We all know that cell is enclosed by a cell membrane, and the next important one is nucleus. All the cells are made up of nucleus, and the next important component of cell is cytoplasm. So the fluid matrix present in the cell is known as cytoplasm. And the next important components, as we all know that cell contains cell organelles. So here, if a cell is going to divide, the new cell must contain the cell membrane, nucleus, cytoplasm and cell organelles. So if a cell is going to divide, which one will be formed first, how it is formed and how the cell is divided. Let us get into the topic now. Okay, so here as I told about the brief introduction about cell, if you take a cell, okay, so I'm taking a cell. In the middle of the cell there is a nucleus. The nucleus contains the DNA genetic material. Okay, so this DNA genetic material in the form of double helix DNA structure, and it is present loosely, like it is called as chromatin fiber. The chromatin fiber is nothing but the double helical DNA structure which is loosely present inside the nucleus like a thread, and here it is enclosed inside the nucleus. And how many chromosomes are present in a human body? That is the question. So if you take a human being, each and every cell consists of 23 pairs of chromosomes. So it is nothing but 2n. N is 23, and here totally we contain 46 chromosomes. Okay, now remember this. Okay, so here human being, inside the cell, it contains 23 pairs of chromosomes, that is a 2n cell, and totally 2 into 23, it is a 46 chromosomes, right? So here it is a chromatin 5. Now this cell is going to divide. Now why cell has to divide? See, for example, there is a cut on your skin because of some of the accident or any other thing. Okay, so if there is any cut on your skin, immediately the skin cells will replicate, they will proliferate very fastly. And if you take a nerve cell, the nerve cell, once it is formed, again it cannot divide. So there are different types of cells in our body which will replicate quite often, and there are many cells in the body which cannot replicate very oftenly, but due to some stimulus they will replicate, otherwise they won't replicate, they are always in resting phase. And there are few cells like nerve cell which will not replicate at all. Once it enters into the cell cycle, again and again it won't enter into the cell cycle. So here, when a cell is going to divide due to any reason, so there is a sequence of steps. If you can watch this video until the end, you can easily remember the complete concept of mitosis, and I promise you that you'll never forget about this concept. Okay, so initially the first step in cell division is G1 phase. It is called as G1 phase. G1 phase is nothing but gap 1 phase. It is also called as gap 1 phase. Okay, so here the gap phase is nothing but when the cell is going to divide, the cell has to be prepared for the division, for mitotic division. For this, the cell needs to undergo some of the steps before going into the division, right? So here in the G1 phase, in this gap phase, several phenomena takes place, several steps takes place. Those are very important steps. Let us see. The first step in gap 1 phase is making of more cell organelles, right? So we all know that in a cell there are many cell organelles present. So if you take any cell, the cell contains different types of cell organelles. For example, the ribosomes are present, the mitochondria is present, the Golgi complex is present. So in this way, if the cell wants to replicate, all the cell organelles also have to be replicated, right? So obviously, and here the first step in gap phase is all the cell organelles will get replicated. That is the first and foremost thing that happens when the cell wants to replicate. Very easy. And coming to the next point, the synthesis of proteins and enzymes. Here, we all know that ribosomes are involved in the synthesis of proteins and enzymes. Why the proteins and enzymes have to be synthesized? If you want to cook a dish today, and if you want to cook a dish today, you will make ready all the ingredients for cooking that dish. Without any ingredient, in the middle of the cooking, you can't run anywhere to get the ingredients. So here, when a cell wants to replicate, it will get ready all the ingredients necessary for its division. In the same way, for the division of the cell, there is an involvement of many proteins and enzymes. So the cell has to be get ready with all the proteins and enzymes. These are the ingredients for the division of the cell. So here, the proteins and enzymes required for the DNA replication and the division of the nucleus, the division of cytoplasm, all the enzymes and proteins get ready in the gap 1 phase. And this is the next important thing that happens in the G1 phase. Okay, and the next important thing that happens in gap phase 1 is repair of DNA. Okay, so why the repair of DNA is necessary? So for example, if there is any change or there is any modification or there is any mistake in the sequence of DNA that happens in the previous cell, the host cell. See, for example, if there is any repair or mistake in the sequence of the DNA is present in a cell, and if it is the situation, the cell cannot replicate the same kind of mistake or error to the newly developed cell. Is it? So here, the thing we have to do is this proteins and some enzymes will repair the DNA which contains some of the errors in its sequence. So that also happens in this gap phase. So it is getting ready for the correct division of the cell. So all this procedure and all this preparation takes place in this gap 1 phase. Is it clear? Okay, shall we go to the next phase, the S phase, right? What the next phase is called the S phase. S phase is nothing but synthesis phase. So here we simply call it as S phase. In this S phase, mostly the synthesis of DNA takes place, which is a very important step in the process of cell division. So here the first step is DNA replication. So here the replication of DNA takes place with the help of several enzymes, and those enzymes are called as DNA polymerases. DNA polymerases. This DNA polymerase enzyme helps in the replication of DNA. As we told that in G1 phase, already the enzymes and proteins required for the division of the cell has been already prepared, and these enzymes help in the DNA replication in the S phase, that is a synthesis phase. If you take the structure of DNA, this is the DNA double helical structure, and if it is the DNA double helical structure, if you see during the replication process, like a zip, how can open the zip of a bag, in the same manner this DNA double helical structure must open like this on one side, and here again a new strand will develop. Okay, so here the double helical structure, again a new strand will develop on both strands, right? In this way the DNA gets replicated. So replicated in how much number? Let us see. So here, as I told you that there are 46 pairs, 46 chromosomes, that is 23 pairs of chromosomes are present in a nucleus of human being. So now in this DNA replication process, this 23 pairs, that is a 46 chromosomes, become double in number. So this 23 pairs become 46 pairs, and totally it becomes 92 chromosomes. So 23 pairs, that is a 46 pairs, become 92 chromosomes. So coming to the S phase, that is a synthesis phase, we can see totally 92 chromosomes present in the nucleus of the cell. So the chromosome number has been almost doubled, and we can call it as the foreign number of chromosomes because there is a 92 chromosomes, 4 into n, that is a 4 into 23. It becomes 92 chromosomes. So the duplication of the DNA that takes place in synthesis phase, and the number of chromosomes are 92 chromosomes. So now let us see what happens in the next phase. The next phase is called as the G2 phase. The G2 phase is nothing but the gap 2 phase. In this gap 2 phase, what happens? See here, already we have seen the replication of the cell organelles, and we have also seen the replication of the DNA. DNA replication has also been completed. So now we require cytoplasm. Now in this G2 phase, the first and foremost thing that happens is increase in the size of the cell. So increase in cell size. Why the cell size will increase? Because of the increase in the cytoplasm. So as the cell is going to divide into two cells, there must be more quantity of cytoplasm. The cytoplasm increases and the cell organelles have already been increased, doubled in number. So the cell size also increases in gap 2 phase. This is what's going to happen. So after this, the cell size will be totally increased, and in this phase the cell is ready to enter into the next phase known as M phase. Let us see what is M phase. So here M phase is nothing but the mitotic phase, the actual phase that starts after the 3 steps. The first step is the G1 phase, that is the gap 1 phase, and second one is synthesis phase, and the next one is G2 phase. In all these 3 phases, the cell has been not actually divided, but it has prepared everything for its division. So in this phase, the cell is at rest, but all the process that are needed for cell division already has been taken place. So these 3 steps are very important, and we call this phase combining these 3 steps, right? So the G1 phase, S phase and G2 phase. These 3 phases combinedly we call it as interphase. So it is called as interphase. Right? So all these 3 we combinedly call as interphase. I'm writing it again. It is interphase. So here this interphase is a very important phase, and after that we get into the M phase. This M phase is nothing but the mitotic phase. We call it as the mitotic phase. In this mitotic phase, the first phase we call it as prophase. So this is called as the prophase. The second phase we call it as metaphase. And the next phase we call it as anaphase. And the last phase we call it as telophase. So these are the 4 phases present in the mitosis. So now to remember these 4 phases easily, so you can say like this. The first one is prophase P, the second one is metaphase M, the third one is anaphase A, and the fourth one is telophase, PMAT. If you remember PMAT, you can remember all the four stages of mitosis. Isn't it very easy to remember? So now, as we know that the cell contains a nucleus and the nucleus contains the genetic material known as chromatin fiber. And this chromatin fiber is like a loosely packed thread-like structure. And entering into the mitotic phase, the first phase is prophase. Let us see what all the phenomena that happens in prophase. So here, if you see prophase, in this prophase, the loosely packed DNA genetic material becomes condensed. The first step that takes place in prophase is condensation. Condensation of DNA, right? So all the genetic material will be coiled and supercoiled and becomes a rod-like structure. It is condensed. So here, after condensation, as we have seen, the chromatin fibers are like this. After condensation, these chromatin fibers become like this. Okay. So here, the first step that takes place is the chromosome getting condensed. Okay, so now when this chromosome become condensed, this is a 2n chromosome and it has been condensed, already replicated chromosome. So now when it become condensed, we have to see the parts present in the chromosome. Here, this part of the chromosome is called as chromatid. This is called chromatid, right? Obviously, we call it as the sister chromatids. These are called as the sister chromatids. Okay, and the whole thing we call it as a chromosome. And one more part we have to see in the chromosome is the middle part in which it connects the sister chromatids. It is called as centromere. So the center part is known as centromere, and other two parts which are present on both sides of the centromere, the protein which is present on both sides of the centromere, that part is known as kinetochore. This is known as kinetochore. So these three parts we have to remember to understand the process of cell division. So the first one is the sister chromatids, the second one is centromere, that is a middle disc-like structure, and kinetochore are nothing but the protein which encloses the centromere on both sides are known as kinetochores. So the actual spindle fibers, now I am going to explain, those spindle fibers will attach to this kinetochore. So now the first step is the condensation of DNA and forms a rod-like structures known as chromosomes. Right? So it contains the sister chromatid, centromere and kinetochore. And the next important phenomena that happens in prophase is, so here, if you see the animal cell, we all know that several types of cell organelles are present, and particularly in animal cells, we can see a separate cell organelles. Those are called as centrioles. Centrioles, and these are also associated with structures known as microfilaments. So in a cell, the centrioles looks like this, and the microfilaments always they are radiated like this. These are called as centrioles, and especially we call it as a centriole microfilament organizing center, right? So the microfilament and centriole organizing center. So now these centrioles and microfilaments play a very important role in the division of the chromosomes. So these two important phenomena that takes place. So thank you for watching this video. Keep updating my channel. Press the bell icon. You will get the notifications, and we will send all the links related to our channel. Do follow that. Please share, subscribe, and share to all your friends and give a nice comment in the comment section below. We always welcome your valuable suggestions. Thank you so much and have a great day. Bye bye.